Oh, yeah. So yes, we do need our lab coats today. I'm aware that maybe you thought we didn't need the lab coat. So if you have your lab coat, please wear it today. I do have a couple of extra lab coats if you need one. If you do not actually have your lab coats today, how many people don't have their lab coat with them? Raise your hand, please. Anybody not have it with them? Cool. Awesome. You can just, because you're the only one that needs to run your car. I think everybody else has theirs with them, right? You think? You can just borrow one today if you want. Okay. You can have to run out there. Like that just seems like a pain in the butt. Yeah. Here you go. Okay. So if you ever forget your lab coat or like you left it in your car, you just forgot to grab it or whatever, I don't mind rolling them out like once in a while. So you guys know it's not an everyday thing. But for exams, be sure you have your own lab coat for exams. I feel like that kind of speaks for itself, but you could run the risk of getting a point deduction if you don't have your lab coat. While you guys are getting signed into this, keep in mind that we're going to be having a lab safety quiz today. So before we even go over lab safety, I would hope that you guys familiarize yourself with what we're about to go over with this, just like it would be with a regular, Kahoots, pre-lab Kahoots. I have decided that as long as you guys are keeping a 50% average or higher on these Kahoot quizzes, I'm going to keep them participation only. However, if we start falling below that 50%, then I'm going to assume you guys really still aren't preparing at all for the labs, which is the whole point of the pre-labs. I don't feel like that's asking a lot, 50%. All right. So 50% or higher, if we maintain that, then we will keep this as participation credit. So even if you got 50% on it, and I don't mean individually, I just mean the whole class gets a 50% or higher average, then that will support everybody and getting full credit every time we do pre-lab. That sound fair? If we fall below that, I'm going to assume that you guys aren't doing any prep work for this, or at least most majority of class isn't, and then we will fall back on what the syllabus states, which is that this is for credit. So I do have the ability to reserve my right. Please don't do that because I don't want to, you don't want to, nobody wants to. It sounds like screwed up, so let's just not do it. So we'll say that. For lecture stuff, I posted last night our what did I say? Critical thinking, yeah. Critical thinking question examples. I feel like a pretty good example of what to expect for our critical thinking style questions on the test. There will be a handful of those on the test. Most of the test questions will be somewhere between the basic, like match your term with a definition type style question, somewhere between that and these critical thinking ones, but you will have some critical thinking ones on the exam. So be prepared for those types of questions. You need to be able to read those questions and pick out the important information that applies to the question and then find the answer that is appropriate for that. Common mistakes, thinking that every single word in the question is important for the answer. You be able to read that question and pick out the important stuff. Other common mistake, not reading all of your choices thoroughly. People will just see a choice that matches a word that they recognize with something and not actually read through it or read all the choices. There might be a better answer. So be sure that you're looking at all your choices. Like if I talk about some type of media that just happens to be selective and differential, and your first choice is that it's selective, and you just collected that because you're like, oh yeah, it's definitely selective, but you didn't see there was a choice that it's both. It's both. It's not just selective. It's not just differential. It's both. Both is the right answer. So keep that in mind. There's not partial credit on any of these either. So be sure you look at that. Now, those questions might not make a whole lot of sense until you have actually gone through the content for all of Unit 1, because a lot of those questions tie in a lot of the content from all of the chapters in Unit 1. And that's kind of the point of those questions, is that we're learning about these concepts to help apply them in real world scenarios. Because your patient isn't going to show up with exactly textbook this, that. You need to be able to critically apply your understanding of the bacteria and viruses in order to understand the difference. One of the things that we talked about in lecture today, if you haven't started chapter 4, that's okay. You'll get to it eventually, I'm sure. But it's a very, very core chapter in what we're going to be discussing going on throughout the rest of the course. So be sure to take your time to understand the concepts in chapter 4. But we talked about the differences between gram-positive and gram-negative bacteria. Now, I'm not going to get into the details of those just yet. We will talk about those details in lab for exercise 3 when we get to it, because you'll learn that in lecture and in lab. But for example, if your patient comes to your ER and has symptoms of meningitis, meningitis can be caused by bacteria, fungi, and viruses. The worst is bacteria by far. And there's several, and I mean several different kinds of bacteria that can cause meningitis. The two biggest ones are going to be Neisseria meningitidis, which happens to be gram-negative, and Streptococcus pneumoniae, which you think causes pneumonia, and you'd be right. It's also the #1 cause of communal pneumonia, but it's gram-positive. So your patient comes in and they've got meningitis. First thing you're going to do is you don't know if it's bacterial, you don't know if it's fungal, and you don't know if it's viral. They present all the same way. That's why it's called meningitis. It's defined by the symptoms, not by the cause. So they have meningitis, probably. They've got light sensitivity. They've got a sore neck. They have a fever. They're lethargic. They might be a little confused, things like that. They come in, and the first thing you're going to do is prescribe them broad spectrum antibiotics without even looking at it. Why? Because bacterial meningitis can be that deadly that fast. You don't have time for the lab for that. But what's the next thing you're going to do? The next thing you're going to do is draw a spinal tap. You're going to get CSF, cerebrospinal fluid, from a spinal tap from them, and you're going to send it to the lab for a gram stain. Because a gram stain isn't just going to tell you if it's gram-positive or gram-negative. We already said Neisseria meningitidis, also known as the meningococcus, and then Streptococcus pneumoniae, also known as the pneumococcus. You probably heard of these before. We have vaccination for them. They might be the two most likely culprits, but meningococcal meningitis is significantly more deadly, significantly. And we are talking about kill you very, very quickly if we are not treating. And that's what we're really targeting with our antibiotics, is that we're hoping that it's not that, but we're going to wait for the test results. We can look at that gram stain of that CSF. We might see gram-negative bacteria. It's meningococcal meningitis. We might see gram-positive bacteria. It's the pneumococcal meningitis. We might see big cells that have nuclei and like mitochondria and all these membrane-bound structures in them, eukaryotic cells. We might see that. That's going to be fungal meningitis. Or we might not see bacteria. We might not see eukaryotes. We might see just clear CSF. You know what that would mean? Viral meningitis. You don't see anything with the virus. So with one simple gram stain, a 10-minute stain straight from a CSF will tell you that. If your patient has viral meningitis, congratulations, they'll probably recover on their own. They probably don't need to be in no ER. But it's better safe than sorry, right? So it's that sort of stuff. So we're talking about the big picture ideas here. Knowing about a gram stain is great because, yes, it's the bread and butter of bacteriology. Knowing the difference between gram-positive and gram-negative, that delineation, they might as well be just two completely different things. That's usually step one with identifying bacteria. So it's our bread and butter with that. But it goes way beyond that as far as practicality and testing your patients and being aware of how this applies to treating your patients when they have certain diseases. But outside of that, when you guys get to watching the lecture, it talks about that a little bit, as well as the importance of understanding the difference between gram-positive and gram-negative as far as like certain environments they might live in and things like that. So when you get the time to watch that lecture, I'll be posting it today. I recommend watching the current one, like I said, because it's going to be, you know, this is what you need to know for the test and not like extra stuff and passing lectures. But I did want to say that because I don't see you guys for a lecture, and all you get is I'm relying on you guys to watch lectures, and I'm not stupid unless you don't actually watch them. So, you know, Also, you know, I can see the view count on the videos, right? Some people don't realize I like check that stuff. But yeah, when I see that there's like 10 views and then before the test, there's like 20, that means only 10 people watch the video. You think I don't know that? Well, I do pay attention to that. But if you're one of those people that you don't watch the video and all you're doing is looking at the summaries or doing the quizzes and you still get an A on that test, man, good on you, like genuinely. But if you aren't watching the videos, if you're just doing the bare minimum just to get by the class and you end up with C's on your exam, I didn't do that to you. I just want you to remember that, okay? So keep that in mind. You do you, but recognize that you're doing you. I'm not doing you, okay? So do what you want, but keep that in mind. So look through your critical thinking questions and be willing to recognize the fact that you're going to need to apply concepts from multiple chapters in order to really assess a lot of these questions and answer them. If you have any problem understanding why the right answer is right, contact me before the test, please. Let me help you understand it before you get to the test, rather than getting to the test and sucking on it and then being like, maybe I should have paid more attention. So don't do that to yourself. Also, when you're doing any of these, and one thing that I don't spend enough time with you guys on this on, if you're doing Kahoots or the quizzes, or you're doing the practice critical thinking questions, it doesn't matter. If you're answering the questions, you need to be thinking about why the right answer is right and why the wrong answers are wrong. Because if I were to ask you, well, it's easy enough to say, this is the question and this is the answer, but why are the other answers not right? And it's easy enough for like simple definition questions, but please try to do that. Like when you look at the critical thinking ones, I promise most of you are just like looking for the right answer. This class is not about just knowing right and wrong, because there are going to be gray areas in there that you're going to need to be able to understand a little bit better beyond just like the black and white, please. So understanding is key. I can't stress it enough. But when you look at those, don't just look for the right answer, because I do give the answer in the comments, but at least understand why the wrong answers are wrong. Okay, it's going to help you a lot. And treat your Kahoots that way too, please. I know like a lot of you guys just clicking through it and then submitting it, and maybe you're not even doing very well on them and you just want to submit your picture, and that's fine. Like I said, you do you, but you can go back in there if you need to redo it and actually make sure you're understanding it well. Or if you don't understand it, you can always email me and ask, I don't understand why I got this question wrong. Look at your quizzes too. I've now opened those up, where now you can look at those and see that you got this question wrong. You need to look up and figure out what the right answer is and why it's right. If you can't figure that out, email me and I will help you with that. So take your responsibility to make sure you're understanding what you're getting wrong on your quizzes and why it's wrong too. Okay, it'll help you, I promise. Just flying through and getting your scores and then taking the test, that's a guaranteed C in this class. Promise. Most students, I hate to break it to you, especially in this section, going to get C's. Okay, be prepared for that if you're not putting work in. All right. So, you can't explain these topics to me, you deserve a C. That's how I feel about it. So, do your work to earn a grade, and you'll earn the grade that you earn. Okay. Having said that, let's go ahead. We'll get started with the Kahoot. And like I said, as long as we're keeping up like 50% or higher on these Kahoots, I'm going to give you guys participation credit. Let's do it. So this one only has 5 questions on it, so it's not so bad. All right, first question. Where should you put prepared slides after you're done with them? So you've made your slide and you are done with them, and now you need to dispose of it. How do you dispose of that slide? All right, so when we have prepared our slides and you're ready to dispose of them, they go in the sharps container. Why? Because they're glass, right? If I put glass in the bio trash and somebody goes to take this, just a bag, and to take it to go autoclave or whatever, that glass can break in there and cut a hole in that bio trash bag, and it could cut the person who's handling that bag. It could cut the bag open, and then the bio fluids could go everywhere. It's disgusting. So even if your slide isn't broken, it's got sharp edges on it that could easily poke through the bag. We're going to dispose of those slides in the sharps container. So that's for safety purposes. This is the hardest question on the whole thing. So don't just beat yourself up over this. But yes, that's where that's going to go. Sharps container. All right. What should you do if you spill a bacterial culture? Sorry, there's no sound on this. All right. So yes, so notify the instructor. This is pretty straightforward. I am going to teach you guys how to clean up the spills on your own, and you'll know how to do that. But just notify me at the very least so that I know what's going on. Just good practice, right? And that way, like if you spill a little bit versus you spilling a lot, there might be a little bit of different procedure going on there as well. Just notify me, let me know. Keep me in the loop, man. Okay, if you have an open wound on your hand, you should notify me, bandage it, and wear a glove. True or false? All righty, so this one's true, also a little bit self-explanatory here. In this lab, starting on Monday, we are working with live bacterial cultures. We're talking about salmonella, we're talking about E. coli, we're talking about staph, we're talking about actual live cultures in this course, okay? They are not the pathogenic strains, except for a few. A few of them are pathogenic strains, but they shouldn't be causing disease in healthy people. If you have severe immunodeficiency, then they would be a problem with you handling them the way that we should be handling them in the lab. If you have an immune disorder, if you are on Humira or other biologics that's similar to that, that lowers the functioning of your immune system, which Humira does if you didn't know that, or if you are pregnant or something like that, then please let me know so that we can have precautions in place so that you are safe in the laboratory when we're functioning. Otherwise, if you are otherwise a healthy person, then you should be fine with the practices that we have in here. You shouldn't even need to wear gloves in here. Gloves, you are welcome to wear gloves if you want to, but I will not provide them to you. If you hurt yourself, like if you have a cut, or if you got a manicure and there was a little bit of, some scabbing going on, even, or your cat scratched you. Man, I had a student one semester that I don't know what the deal was with him and his cat. He was always getting scratched by his cat, or whatever. Then you'll want to wear gloves just to protect the bacteria from getting in there, because you don't want to set yourself up for the infection, right? We want to keep you safe. If you have an open wound, or if you have something that's relatively fresh, we can always put a Band-Aid on it and a glove just to be sure. So I will have gloves available for you for that if you need it, but otherwise you aren't required to wear gloves in here, and I won't be wearing gloves typically. So keep that in mind. The only thing you really need to wear in this lab is your lab coat and closed-toed shoes. And even to me, that's a little bit open for interpretation. You'll see me wearing like flats where I'll have most of my shoe, my foot is open, exposed, but if the toes are covered, that's what I'm looking for. And to me, Crocs don't count because enough of the liquid can seep in there, so that's gross. So if you were to spill culture on there and everything like that, that's the idea. I'm not concerned about like, steel-toed boots and stuff, but I do want you guys to at least have your toes protected from spills and potentially broken glass and things like that. People break tubes in here all the time. Like that's a pretty big hazard. So closed-toed shoes and lab coat is the only thing you have to have. You want to wear goggles? Don't let me stop you. Actually, I think that's the next question. What two things are absolutely required? Absolutely required. Remember what I said, guys, if you forget to bring your lab coat with you, just let me know. Have your lab coat, or I'll take points off for those days. So lab coat and closed-toed shoes. Good job. You may wear goggles. You may wear gloves. They are not required. True or false? Food and drink. I mean, relatively, like you could pick it up and drink from it. Food and drink are allowed in the lab if you store them under the bench. True or false? I also feel like this should be obvious. This is false. We are working with actual bacterial cultures. Please, please don't bring like even your Stanley cup. Like it has a straw on it that you could just drink out of. Please just put that at the very bare minimum in your bag where it's secured. I prefer that if you have a drink like that you could just sip out of, that you take it and eat it in the hallway. You're welcome to step out in the hallway and have sips of whatever you need, whenever you need. And even me, like once that we're starting with our wet lab stuff, I will have my drinks out in the hallway. I drink a lot of unsweet tea, like constantly. I don't like sweet tea. I like unsweet tea. Everything weird, and that's fine. But that's that's my beverage of choice. I drink a lot of it, and I usually leave my cup out in the hallway, and that. So please do the same. You're welcome. Like I said, take a break and go out there and have a sip of it. But I don't want anybody spilling their culture on a drink and not realizing it, and it's gross, right? So this place is gross. Please treat it like it's gross, right? You will see what I mean, right? So that's it for our safety pre-lab quiz. Typically, our pre-labs will be about 10 questions, and they will be about what we will be learning about. So please just at least peruse that information. And you can, I'll tell you this, it's open note. So you will have your lab books there too. And in case you guys didn't know, our lab books are today, now they should be next to your microscopes underneath the lab bench there where you guys are sitting. So everybody has their own microscopes, everybody has their own lab manuals and all of that. So yeah, if you want to move to one of the open seats, Omar, that'll be a little easier on you. Also, book bags. And most of you guys are following this rules, it's not a big deal. Just be sure your book bags are like out of the walkways, straps, bags themselves, either against the wall or under the bench. So that when I'm walking around, and I will walk around, that's very engaged a lot while you guys are working, and you guys will have questions. I don't want to trip on your book bags, and I don't want you tripping on each other's book bags. That can become a liability. So be sure those are just stowed away appropriately. Anyways, your lab notebooks. So on Monday, we will be doing exercise 1, for example. So you would find exercise 1 is on page 3. These first 2 pages are our lab. You could have had this open during the quiz and then see if you found the answers here. Not that it was hard, right? That was pretty easy, but anyways, exercise 1 is here. This is like our lecture type content for exercise 1. These are your stepwise procedure. Now, I will go through the procedure myself. I will, at least for sure in the beginning, actually do the experiment. And you can either watch me do it and follow along while I'm doing it, or you can watch me do it and then do it afterwards on your own, and I don't care. But the steps are here no matter what. So if you get lost on what you're supposed to do, you can follow these steps, and if for some reason these steps are confusing to you, you can ask me and I'll come over and help you. Anytime that anything happens in this lab where you're confused about it, you're always free to ask questions, and I will never treat you like, why are you asking questions? Or I don't know, what do you think? Or anything like that. I don't do that, and I don't approve of it. So if you have, I feel like if you have the nerve to ask a question, you deserve the proper answer. So I try to treat you that way the best I can. Having said that, if you break something in the lab or have a spill or something like that, tell me about it and I won't get mad. Don't tell me about it and watch how quick I change, because I'm not going to be happy about it if you are spilling something and you don't tell me. Break my microscope and you don't tell me. Break a piece of equipment and you don't tell me. You don't belong in a hospital or in a vet clinic. I'm sorry. Grow up. Okay. Take responsibility for your actions. So you tell me about it and I will be there to help you every way, and I will not be mad. If you break even the whole freaking microscope and you knock the whole thing, I know I wasn't supposed to have it out yet, blah, 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 but I knocked it off. I'll be like, I'm not going to lie, my eyes are a little frustrated. But I'll be like, no, we're going to help you out. You're not going to get kicked out of the lab, out of the class, or fail or something like that, right? I'm going to help you out, we're going to figure it out, and we're going to solve the problem together. So keep that in mind. You make a spill in the lab, you tell me about it as soon as you can, and we will figure out how to make sure that we're cleaning it up correctly. You probably will know how to clean it up correctly. Most of the time it involves dousing it in disinfectant. So that's not a problem. And we'll get to that when it applies. So you are welcome to use those during the lab quizzes. Okay. Keep that 50% or higher. All right. All right. Okay. So let's go over the lab stuff. So we figured out where our lab manuals are. These stay in the lab. They will stay down there by your microscopes. Everybody gets to use them. You're welcome to write on these with dry erase markers and if you need to. If you want, even put a marker in here. If you want to print out your own actual paper ones and write on them with your pen, keep that in a little pocket for yourself. You're more than welcome to. I'm not going to print them out for you. All right. So if you want to, you're welcome to. Otherwise, these are just fine. So on these, like I said, you'll have the instructions for the lab we're doing. If you want to write notes up to the side and then take pictures and have that be your notes, that's do whatever you want. Just be sure you erase it before you're done and you put it away. Okay. Mark off the steps as you go through them. Just be sure that when you're done, you erase it. Rub it with some paper towel. And if it gets smeary on there, or you can't clear it off completely, use a little bit of disinfectant and then you wipe it down. Okay. If you spill on this, please, a little bit of disinfectant, wipe it down. Okay. All right. Then we have a study sheet. Usually for most of them, they'll have a study sheet associated with them. It's like the big take-home stuff from when we're learning. And then the actual sheet that you would be filling out for credit. We don't have one today because we're just talking about lab procedures. So starting on Monday, we're going to start taking pictures of what you put on your answers and you upload it into your in-lab worksheets. You guys already know how to take pictures and upload because y'all been doing that with your screenshots. So you know how to do that by now. But it's the same thing. You'll just take a picture with your phone of this whole page, upload it into the in-lab uploads for that exercise. And then I actually grade those. So it is graded on accuracy as well as effort. And you do get partial credit for being in the ballpark. So like if I'm asking something and you didn't get it right, but it is still in the discussion of what's appropriate, then you'll get partial credit for that. There's a rubric for these that are associated with the assignments that I will be using. If you just decide that, you know, there's no shame. If you like, I don't have the time or I don't want to stick around and do grand rounds or whatever, which we'll talk about what that is. But you just don't want to do it, or you just want to answer a question. I don't care. Like leave it blank, and you know you sacrifice those points. Or don't make no decisions. So whatever. But that's what you'll take a picture of and upload for your in-lab grade. Yes. Yeah. So you can just upload the picture straight from your phone. Yeah. You guys can do that with your screenshots for that too. Like whatever your screenshot is, if it's a JPEG, or if you're taking a picture of it with your phone, some people just take a picture of their screen with their phone. That is fine too. You can just upload that straight up. You don't have to convert it to any other kind of file. It'll let you upload whatever. So if I have a problem with whatever the file format is, I'll complain about it. You'll hear about it. So I usually have not had a problem yet. So just be clear. So yeah, if you were to go forward to like exercise 2, for example, we have our first one that has a grand round. And you'll see here it says, fill the chart out for each table. Noteworthy colony morphology or cell shapes or arrangements. So we're going to learn about that stuff when we get to it. But you basically are going to go to each table and write down some stuff you see at each table for each table. Everybody's going to have different organisms. And so it's important that we go around and see the differences, not just for learning experience, but also because whenever you guys are taking the test, I might have a slide up here with cells that didn't look like your cells. You know, it might be a different shape, there might be different arrangements. You want to have an idea of what each of the differences could possibly look like. It could be because y'all are going to suck at making more sayings in the beginning. Like most of you aren't going to get it right the first few times. And so you want to see what one looks like that was done right, and what a positive looks like and what a negative looks like. So you want to be sure that you're actually seeing that stuff and engaging it with different ways, so you can know what to expect for different settings and scenarios and whatever. That's the point of the grand rounds. Also get you guys to kind of talk to each other. My experience with the hybrid class is that you guys are the least communicative by far. Like a noticeably different environment in this class than in my other classes where we're all chatty and talking. And this class is usually like a word. It's like the quietest class. Nobody's laughing at anybody's jokes. Nobody's engaging. Nobody's talking. Nobody's afraid to ask questions and everything. And that's a detriment to you guys. But it's because you guys don't interact with me as much in lecture. It'll get a little easier as we go through and you guys get to know me. You realize I swear a lot and I have a problem with that. And and I will try to apologize. I really try not to, but it's just like I don't think about it and just like in my mouth. So it's never intentional. But yeah, I have a potty mouth. I'm not gonna lie. So things like that. Like you'll see, it's a lot more laid back than probably what you're used to in other classes. And I hope that works to your advantage later on. Just don't be afraid to ask questions. I don't want that quietness of this section, which it just is this way. It's not your fault or anything. So I don't see you as much. But I don't want that to drag you down somehow. So just don't be afraid to ask questions if you have it. Okay. But whatever. So every day, daily, I have what do we say in our lab? If you guys actually watched the lab video and how I actually am inclined to believe that many of you didn't, then that's okay. You should be coming into lab, squirting down some disinfectant before you even sit down. Work your area and then wipe it down. I know a lot of people didn't do this. I will say this in almost all of my other sections, my in-person sections. I don't know what it is. It's like somebody sees somebody like do it, and they're like, "Oh yeah, that's the thing we do." So when I have people that come in a little earlier and have watched it and have already done it, and you guys don't see it, and maybe, that's what it is. But yes, my other sections, I can look through to you. Every single one of them will have cleaned their bench today before I even mentioned it. But they watched the video and they saw somebody else do it. Almost none of you did. Who raise your hand if you cleaned your bench before you sat down? Interesting. My entire class in the last section did. That's what I mean. Okay. So already y'all falling behind. And you think that's not, but that's where it starts. And that's what I'm talking about. Okay. So that is in our lab safety video that you watched, right? So you should be coming into lab, disinfect, because you're going to assume that the person that sat at your seat before you spilled salmonella there and didn't clean up after themselves. Because I'm not cleaning up after you. I'm not your mom. It's your responsibility to clean up before and after, right? So you come in, clean up because that person spilled salmonella there, just assume it. And then you can sit down and do the Kahoot or whatever it is. And we're not doing anything super nasty, so don't freak out or something. Today you're probably fine. And because we're going over clean before you start and clean when you're done, the people before you did clean when they were done and make sure of it. So you're fine. But keep that in mind. You need to be cleaning before you get started. Then we will do the lab, pre-lab Kahoot like we just did. You can do these either way if you want to. I really prefer that you disinfect first, though. It's just gross in here. And then we will jump into the lab concepts where I will do a mini lecture, kind of like I am now. And then we will jump into whatever we're doing. I usually am going to do a demo. Like I said, you're welcome to either follow along while I'm doing it or watch me do it and then do it yourself. Your steps will be available for you in your lab book. And then we have that worksheets that you'll do on the dry erase, take a picture and upload into your in-lab worksheet assignment. All right. That's all on Canvas. At the end of lab, when you are done, you do not have to wait for the rest of the lab to finish. Whenever you are done, you are free to go. Okay. But before you are done, you will disinfect, wipe it off. Just squirt your area. And if, let's say you get in and like you're the first person at your table, if you clean the whole table, that's fine. Just as long as everybody's spots got cleaned before we got started. Okay, you can do it for your team if you want to. I don't mind. But as long as it gets done, and I'll kind of pay attention. I'm not going to be like super over your shoulder, making sure you're doing it all the time. But if I catch that somebody is regularly not doing it, I'm going to dock you points for class. Okay, it's a lab safety issue. It's gross. It's like legitimately gross in here, and just always treat it like it is. All right, so disinfect when you're done. So that's the things you should always be doing just to keep like clean and safe and everything. Disinfect when you come in, then do your whole thing. Disinfect when you're done. Wash your hands before you leave the lab. And just to be clear, in case there's somebody out there that isn't sure about what I mean by washing your hands, this is what washing your hands appropriately would look like, okay? Turn on your water with your nasty old hands. Get a couple of soaps. You can wet your hand first if you want to. I don't care if you do it before or after, but just be sure that you get soap, water, suds it up. This is not enough, right? Suds it up. You're supposed to sing happy birthday or ABCs or whatever you want to do. I'm not real particular about it, but do give a good sudsing to your hands. Try to work it into your nails a little bit to get some sudsing going on so that you can get some nasty to bring it home to your babies or whatever. Wash your hands. Give it a good zhuzhin under the water stream. Wash all that soap off real good. Then do not reach and turn off the water like that. Y'all been doing it, and I know you have. Dry your hands to nice and clean before you go toss this. Turn off your water, then go toss it. Don't be touching that nasty handle with your clean hands, right? You know this, but actually practice it, especially in here. And then even if you know if people aren't even washing their hands right either. Okay? Not worried about it now, but this place is nasty, okay? Already, all this, old bacterial cultures, just chilling out here. It's gonna be like this constant, and it's only gonna get nastier. They're gonna walk in here and they're gonna smell it. And just trust me, it's gross. So that's why I want to keep clean. Don't take it home to your babies. Don't take it home to patients. Gross. Okay. So wash your hands. All right. Other lab stuff. Everybody clear on like your day-to-day responsibility? Cool. All right. Incubators versus fridge. Now, technically, yes, these are fridges. They even said one, right? I want to be clear. These are our incubators. So if I tell you to put your samples in the incubator or get it out of the incubator, it's usually like that. Okay. And this one is Monday, Wednesday lab. You guys, and we've got Tuesday, Thursday, pretty cool. You guys know what's going on and what's going on there. These are kept at 33 degrees Celsius. I don't know the exact conversion. It's around 86 degrees Fahrenheit, something like that. Not body temperature, not body temperature, but it is warmer in here than it is in the room. Okay, just to help encourage your microbes to grow. Body temperature typically is going to be 37. Room temperature is 21. Just to put it in perspective. Okay. It's kept at 33. You can see I opened the door and it's already down. And so it's that quick. So please don't leave the doors open. Put your stuff in. Close the door. If there's a bunch of people waiting in line, you can leave it open. But just don't leave it there like your last one. Ready. So we'll put your cultures in here. This top two shelves is my Monday, Wednesday morning. You guys will be this shelf. So if it says Monday, Wednesday, 1 p.m. It's you guys, this shelf and this shelf. Okay. They stack plates up in here. They've got the whole table plates stacked up in one nice little stack. You're welcome to stack the plates. If you really just want to put them out separately, that's fine too, but please don't take up more than your two shelves. This is my shelf. You can't go over too much space because I have to share my incubator space with my manufacturer. That's their lab over there. You have noticed it's not really functional yet. They're using my space right now. So we can't like just keep it all nice and consolidated. The other reason that I want you to keep it consolidated, not just put it wherever. Because you might be like, oh, I remember I put it on the top shelf, but I know I should have put it there. But if I have to come in because of a weather day, and I want to save you guys the samples from overgrowing or being contaminated or something like that, I'll come up here. I have 4-wheel drive, and I'll come up here and I'll take out your samples and put them in the actual refrigerator, which is cold, which is this one here, this little platform, so that they don't ruin it. So we don't have to redo the lab or miss out on it or whatever. So I'll come and do that, but it's way easier for me to keep your samples organized in that tiny little fridge if you guys have organized it originally. So do that and label them. We'll talk about it a little bit. So that's the incubators versus the fridge. That's the whole fridge. You will open the door. Yeah. If you want something to grow, it goes in the incubators. If you want something to stop growing, it goes in the fridge, right? All right. Refilling your own bottles. So we're going to be using water a lot in our stains to like rinse the stains off in between our steps and everything like that. You're going to run low on your water. If your water needs to be refilled, you fill your water bottles up here where it says fill water bottles. This is deionized water. It's supposed to have a light on it, and I don't know what happened with that, but it's not working the way it's supposed to. So I have to call the Culligan guy and have him fix it. But until then, what I want you guys to do, if you need to refill the bottles, please do it on your own. So I'm not going to refill them for you. If all of your water bottles end up empty on your table, I'm just going to leave them like that, and you're just not going to do your stains properly. Because we're not babies. You can fill a freaking water bottle. Just let it run for like 15 seconds, and that usually gets the deionized water flowing out properly, and then come fill it. Do you guys know why we have to use deionized water? Anybody have a guess? Anybody read through chapters 1 and 3 or watched the lecture yet? Anybody? So we've talked about the different kinds of stains, right? And the positive and the negative stains. Well, for something to be positive versus negative charge, interaction with the cells, which have a negative charge surface, in order for that interaction to happen properly, we can't have interference of charged ions. And so that's why we have to use deionized water. So please refill your water bottles with the deionized water. It'll save you a lot of trouble later on. So use this for your water bottles, okay? If you need to wash your hands, use the regular tap. You're welcome to use my tap or the tap back there when you're washing your hands so you don't have like a big old line just at that one, okay? Say mine, it's not mine, it's out. But it just happens to be by where I sit. So soap, this is hand sanitizer. The hand sanitizer by the door, it still isn't working even though it's complained on it. It's full, it just isn't working. So you're welcome to use this hand sanitizer. Hand sanitizer is effective as long as you get enough and you're actually rubbing it really well all over your hands. So that's fine to replace hand washing if you want to do that. That one by the door isn't working, like I said, but there's another one right outside of Dr. Shearer's lab that is working. So if you want to use that one or this one works. Maybe this, to me, one little squirt isn't enough. I usually get 2. and then rub it. It's wet, and I know it is. Just put a lot of it on there, and it's gonna be way more effective than just a tiny, tiny bit that you're just hoping will spread out. Just trust me, okay? It works the way that it works because of that concentration that we're using. Okay, so all right. Yeah, refill your bottles. The disinfectant bottles, which are the red bottles, like we do disinfect with the red bottle stuff before you come in, when you first get in, and then when you're done. That's in the carboy back there. I know we've moved it. It used to be on this red tray here. You just have to put it back on the red tray. But where it says refill disinfectant, this carboy right here, it also says refill disinfectant bottles here. This is your disinfectant. Just come over here, turn the little thing, and it'll go. If a lot of people are doing it that day, they might get some suction built up in there. So just loosen the lid a little bit, and it'll let the airflow, you know. So refill your bottles, please. Racks. We're going to be getting out samples of our bacteria and doing multiple tests with them. And you might have one person at your table doing this, and another person at your table doing that, and whatever. So having one rack at the table, which you will have, might not be enough. You might want to have more space to work with. Anytime that you guys feel like you want more racks or want to divide up the work or put, you know, stuff that I have inoculated over here, stuff that needs to be inoculated over here, and stuff that I need to read results on over here. Get as many racks as you need to do that. I will only give you one at your table, but if you want more, you can come and grab whatever you want from any of these racks that are over here. Anything from this shelf down here is fair game. Okay, just be clear. These won't be here. They'll be the ones that are at the table. You just won't have that. But anything that's down there is fair game as far as the racks we put up with the tubes. If it's up here, please don't take it from up here. Okay. Who's looking up here for that? So whatever. Don't take some up there. Even if it has a label on down here, because it's not. Okay. Take whatever you need from down there. So you guys don't feel like you have to work in that one little rack, and it can just be a lot eventually. Drawer contents. Let's go over what's in your drawers. Everybody's responsible for the contents in your drawer. If you get to your drawer and something is missing, or you have a concern about something in your drawer or whatever, somebody just ruined something, then please let me know, because then I'll know who did it, right? The person before you. So in your drawer, you have your staining tray. And yes, this thing gets to be a mess, and you're pretty much going to get stains on your hands and stuff. If you have nice, new, fancy nails that you got done, wear gloves, okay? So a lot of times that stuff will come off with alcohol, but I don't like to keep that alcohol around for that purpose, please. So keep that in mind. That'll be your responsibility to make sure you're not doing that. But anyways, you have your tray. This is what we're doing our slides on. Don't worry about how it works now. You have in there this thing. This is a fan. Trust me, you're going to be thankful for it. Our slides have to be air dried before we can heat fix them. So that's there to help you dry that stuff out. To put them away, you just twist them up until they just keep twisting them like your sun visors on cars. And then just put them back in the little pocket and throw them back in. And we have stains. We should all have 11 bottles. I've gone through and make sure you have enough. Some of them might be on the lower end, like acid alcohol was on the lower end. When we get to exercise 4 where we use this, then if you run out, just let me know and I will get you more. I've made more. I just haven't refilled the bottles for that. It eats through the rubber really easily. The acid alcohol does. So I don't like to fill it too full. So last, we have this tray of nondescript items. You should have a Sharpie, at least one Sharpie. At least one China pencil. It might be the old school China pencil. It might be the twist up kind. A dry erase marker. A lot of people have like 5 of them in there. But at least one dry erase marker. These are absolute trash, okay? So they'll run out. And if yours runs out and you want a new one, then just let me know. Just toss it. And there are some up here. You're welcome to just come grab them. It says dry erase on there. Run through as many as you need. You're not throwing them away too often. I promise they just are that bad. So as soon as we can get through these bad ones, I can order new, nicer ones. So don't hold back on it. If you feel like it's getting low, just toss it and get a new one. Okay. The ones that are blue, they have a little eraser at the top. So if you want to keep one in there because it has a little eraser on it, you're welcome to keep it just for that. The tongs, we'll talk about what those are for later when we need them. These are this is a slide holder. This will hold your last slides when we're putting them over the flames so you don't burn your fingers. I'll show you how to use those when we need it. Tape, these are for labeling your tubes. You don't need to label your plates with this because our plates are disposable. We don't reuse the plates. But you will need to label your tubes. We do reuse the tubes. Don't write on the glass. It rubs off really easily from the glass. So you'll write with Sharpie on the actual tape and then put that on your tubes. I'll show you how to do it when we need it. Then we have our inoculation tools. We should have two. A needle, which is pretty self-explanatory, and a loop, inoculation loop. There's a couple styles of the inoculation loop. People tend to be kind of harsh with the loop end of the loop, so they might break. The loop part might break. If you notice that your loop is broken, then you would need to let me know, and I'll give you a new loop. It's not that happens, so don't feel bad about it if you do it to it. Just let me know, and I'll get you a new one. Okay. But you need a loop and we need a needle. The one we're using the most will be the loop, so it gets some wear and tear. Okay. So like this is what we'll be doing. With these ones, they're like all one piece like this. It's got a broken loop, which this one does. It doesn't go around all the way. It's got a break in the middle of it. Throw it away. I'll get you a new one. We have some new ones that I just ordered that I can replace the loop on them, but they haven't come in yet. So for now, we have kind of disposable ones. Here you go. Oh, thank you. All right, so that's the drawer contents. Then the drawers that are on this side of every bench. So this one and this one, or 4 and 1, or this one, or that one and that one. They all have one other thing added in them. That is the striker. Not everybody has a striker. Only 2 seats at each table, one on each side, have a striker. This is how we'll be lighting our Bunsen burners. Using almost every single day, and you'll be running most of the time. This isn't like play baby chemistry time where you'll sometimes be using it, and you know, Dr. Kim comes and lights it for you and gets frustrated. This is you guys are probably going to need to learn how to actually apply these things. Okay, and I think that you know how. I want to show you the difference. If I do this just with one hand, and I'm just doing this like kind of chill or whatever, you can see how frequently I'm going to get a spark on here, which is next to nothing. I hold it with one hand, my left hand, my non-dominant hand, and I'm holding it nice and tight with that hand. Pull the actual striker portion where the flint is, and it should have a flint, which is this black piece that pokes out here, okay? If you don't have a flint on yours, or it's like almost to the silver, please let me know and I'll replace it, okay? But that flint part goes on the actual, this should be rough. Some of them are pretty worn down. That's okay, we can still make it work. But anyways, that flint part goes on that. And what I do with one hand hold, and then with my index finger, push that flint into this as hard as I can. And with my thumb, push away so that I'm pushing as much tension as I can. And now you can see, look at that. We're getting a lot more spark. Promise if you think about it that way, you're going to be way more successful at lighting these. I cannot tell you how many minutes people will spend just going like this. Well, I'm getting the sparks now, but just doing that, shoot for like the good first 10 minutes of the lab. Don't waste your time. Take the time to actually hold it properly and get that going so you can light your Bunsen burners correctly. To turn your Bunsen burners on, we aim the, now let's go over this again when we use them again. But this, the more we know about it, the safer it is. You'll aim your actual like, what do we call this? The handle knob, yeah, the knob, yes, the knob. Aim it down the line to turn it on. Some people will go like this. It's off again. Just off again, okay? This is the maximum right here, okay? Do not adjust your gas flow with this. This is not how you adjust your gas flow, okay? Just treat it as on or off. This is on, this is off. There should be not anybody in between, okay? So on, you'll hear it. If it's nice and quiet in here, you can hear it. But when people are talking and doing stuff, you won't be able to hear it. So if you aren't sure, you can lean in or you'll smell it. It's natural gas, so it has stuff. I don't know if you guys know, but natural gas doesn't actually have a smell. They add something to it so you can smell it. So don't freak out because you see me standing here talking about it with the natural gas going. Like it's not going to flame up this big old ball of flame. There's enough air in here that it's not going to do that. So it's fine. Don't freak out about that. To light your Bunsen burners, do not do this. This is just going to put out your flame. Okay, hold it to the side, at the side of it like that. And then hold it like I told you with the 2 hands and get some good spark going. Sometimes it won't light up the way you want to. See how it'll light for a second and then go out? And it takes a little bit of finagling. It's really not behaving today. Which I get, like, usually I get it in like 1 or 2 cracks. There's just days, man. But anyways, this is adjustable, okay? So you can turn your gas down by turning this little knob down here. This is how you control your flame. See how nice and cute it is? I like a good roaring flame, okay? If you keep rotating this guy, so it's like it goes down, 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 it shouldn't need to be any higher than this, then this will eventually come out. And the gas will pull out of the bottom of this, and it will be on fire, okay? And literally you'll have fire out of both sides of it. And if you're freaking out, you do that, and it might happen, okay? If you're freaking out, you can just turn it off, but if you're still freaking out and you still are scared because you're near it or whatever, then what you can always do is come over here, and big old red button that says emergency gas shut off, and it shuts off, it should be shutting off the gas. It's not off, is it? It should be shutting off the gas for the whole lot. I think that they're doing work next door, they have this change. Okay, that might not work. But it's supposed to. I'm actually going to notify them of that. That's actually really, I think it's because they're working next door that can rewrite gas lines. So that might be just because they work for now. But that's fine. Anyway, normally you'll hit this button, and anybody is welcome to hit the button. And it might work at other tables, and it's not at others. I'll look into that. I'll definitely look into that. But it's actually tested before. Anyways, hopefully I'll get this fixed. I'll have them one night. This should shut it off. You guys are welcome to hit that button, and if it doesn't work, then push scream and let somebody know. There are fire extinguishers. There's one back here, and there's one up by the door. So if you do need to reach for that, you could always reach for that too. Okay. Don't wait for me for that stuff. Okay. If there's an emergency, just act. All right. Anyways, yes. To turn this off, if you were done with this, you would just turn it back where it was. I'll show you how to do that in a moment. But you can also adjust your flame with this thing here. Look at the difference. See how nice and loosey-goosey that is? I don't care for that personally. Like I said, I like a nice roaring tight flame. The smaller that little triangle is in the middle, that's nice and concentrated flame there. Then you will be able to take your inoculation loop or whatever and be able to sterilize it and put it in the part of the blend that is above the inner cone. Okay, so you want to be in the flame, but you want to be above the inner cone. So that inner cone in the middle, let me show you the difference. You guys see this? How we're getting a nice orange all the way across that middle part that's in the flame? See the difference? So you want to be above that cone, and it'll tell you, right? Because we're going to be sterilizing these things. You'll have to get glowing orange. You want to get your whole fire completely glowing orange all the way across. Now, I don't think I need to tell you, but if I do this, you don't touch it to your hand, right? Don't touch it. Okay, we're talking glowing metal. Please do not touch it. Okay, let it cool for 20 to 30 seconds before touching it. At least, okay, 20 to 30 seconds before touching it, if you needed to touch it, or touching it to your cultures, because if it's that hot to you, it's that hot to bacteria. If it's that hot to bacteria, you're just killing your bacteria. You're not going to be able to spread them from place to place. You want to let it cool and then use it. So once it will be cool, then you'll be able to touch it and be able to touch it. This is cold, literally cold now. So it cools off pretty quickly, but you want to be sure that you're sterilizing it appropriately and know how to use it. So you again when we come back next week. So once you're done with the gas, all you do is turn it off. There's nothing else special you have to do to it. Okay. So that's that. Month burners. And that's everything in the drawers. Any questions about any of that stuff? Please keep what's in your drawer in your drawer. You'll notice that the strikers have a number on them. That's the drawer they belong in. If you borrow something from another drawer, if it is because your drawer ran out, like ethanol, that goes pretty quickly. So if you run out of ethanol and you borrow it from another drawer, let me know so I can refill your bottle. It's okay if you borrow it from the other drawer, but let me know so I can refill it so that the next class doesn't have that problem. Okay. So keep me up with that and I'll refill your stuff with you. It's okay if you run out, let me know. I'll refill it. You didn't do anything wrong. If you did do something wrong, you did something you weren't supposed to do, just let me know. Like I said, I wouldn't be mad as long as you're letting me know. Literally 1 year for the exam, I had a student that we had cultures sitting out, they were liquid cultures, and she was tipping it to see, some of them will solidify, they're positive or something, or they might not, they're negative. She was tipping it to see if it was liquid. She spilled it everywhere, she just left it like that. And then nobody told me about it. Nobody told me about it until the very, very end, like the last straggler was like, hey, did you know somebody spilled this? So I'm not going around checking every station within 10 seconds. And then I found out from other students, like, yes, you know, like I saw so-and-so spill it or whatever. Guys, come on. I'm going to say it one more time, do I? You don't. Okay? You spill it, tell me, you're not in trouble. You spill it, you don't tell me, you're going to be in trouble. Okay? Just let me know. I'm here to help you, man. I'll tell you how to clean it up, right? Or I'll even clean it up for you if you don't know. It's okay to admit you don't know. So but I'm here to help you with that. All right. Microscope rules. We're not using the microscope quite yet. And we'll go over the parts and how to operate it and everything when we get to lab 2, which is going to be Wednesday next week. But when we get our microscopes out, because I know you guys are going to be doing it probably before you're supposed to, do not ever get your microscope out on your desktop if there's a Bunsen burner going. Okay? If there's a Bunsen burner on at your desktop, do not get your microscope out. I don't care if you have to wait till the other's off your table. You wait. Okay? If you're going to be running power cords across the table, I don't want you burning that and causing electrical fire on top of regular fire. So when you're lifting your microscopes out, you lift it by the arm. That's just the spine of it, but it's called the arm. And put your other hand under the base. Okay. Your microscope should be set up like this. Your stage should be all the way down. How do you know if it's all the way down? You can either push it to make sure it's down, or use the course adjustment knob. You can actually see it moving up and down, right? So it should be all the way down. Your objective lenses, which are these lenses here, this thing rotates. This is called the nose piece. It has a whole other name. Okay, these are your objective lenses. It should be on the lowest one, which is your 4, to get the most space in between the lens and the stage. Okay. When we are looking at the microscope and using it, I'll go over how to do it and everything appropriately. But you will start always on the lowest setting, always. Find your thing. I'll show you how to do it when we need to. Then switch the next one up. Find your thing. Switch to the next one up. Find your thing. Then go to the highest setting. On the highest setting, the 100x, you will always be using immersion oil. Okay? Always. This is where I'm saying, if you guys have taken a class where you've used a microscope before, and you have not used immersion oil yet, you have probably not used the 100x, or somebody's not teaching you how to use it properly. Okay? 100x always uses immersion oil. I'll show you how to do it when we need it. I'm not going to show it right now. Here is something that is an absolute hard and fast rule for me. If you see somebody put their microscope away like this, the stage is all the way down, but they're on the wrong objective lens. I'm not going to be too mad because it happens, you know, if you're doing it occasionally, I'm not going to be mad. If they're doing it regularly, like if you're always getting your microscope out and it's always on the wrong, it's not on 4 and the stage is not down all the way, or it's a hot mess, there's just stuff all over it or whatever. If they're doing that regularly, tell me about it. Okay, I know people make mistakes sometimes, it's not a big deal. But if they're doing that regularly, I want to know about it so I can talk to them and make sure they know how to put the microscopes away appropriately. Okay. But there is a hard and fast rule that I have that last semester, I was so hot about it. I almost tested people on how to freaking use a microscope and put it away properly and test them like actually like part of like half of their tests. So I was so mad about it. Please do not ever do this. I'll straight up fail you on my course past a certain point. Okay. If you find that your microscope is set up like this, first of all, I also want to be very clear about this. If I ever see you guys using your microscope like this, where you're watching where your slide is and bringing your highest power lens and then bringing the slide up to it like this, I will walk up to you and show them how you use a microscope. If that's how you're using it wrong, period. Okay, don't ever let me see you do that. Okay, you're risking running into your slide here and cracking the slide. You could have glass shooting into your eye from that. You could crack this lens. We're talking these lenses in the 100x are like $2,000 just for this freaking one lens. You're paying for that, guys, out of your own pocket. I'm not paying for that. You are. Keep costs low for people in this class. So don't do this. This is going to piss me off really fast. That's enough to make me want to call you out. But if I see your microscope put away like this, and it happens, man, last semester, it was like every section, at least one person put their microscope away, completely set up still. On the 100x, stage up, slide still in there. You do that once, and I will have a talk with you, okay? It's a warning, it's a talk. Don't do it again. This is how you put your microscope away properly. You do it twice, you're cleaning everybody's microscopes in that lab at the end of class. And then you will stay here and do it late. If you can't stay late, I will make you do it on a different day, You do it again after that, you were talking about points deducted at that point too, probably from the class. But if you keep doing that after I've already walked you through and like punish you, which I shouldn't have to do as an adult to another adult, do it again, I'm just gonna fail you from the class. I never had to do it, but I came real close last semester. But people just kept doing it. But they're gonna be different people. So then how you think you learn from other people's mistakes, but they just were not, man. So just don't do it, please. And I'll help you out. If you don't remember how to put your microscope away properly, just ask, okay? Like, do I put this all the way? Like, I don't remember. I know I'm not starting a slide on here, but no problem. Okay, so what you're gonna do is, and I'll walk you through it. If I have to do it every day, I'll do it every day. But please don't ruin my equipment, okay? Because the way I see it is, if you're somebody who is consistently neglecting this equipment, I don't want you anywhere near my family, period, right? You can't take care of this. This is only worth like $2, $3 grand. My family and my dog's lives are worth more to me than this microscope is. You better believe it. I don't want you anywhere near my loved ones, okay? So you can come back when you're ready to be an adult. But just to be clear, this is like one of those things that I'm pretty, like this is one of those times where I will actually get like heated, okay? This. Now, if you had taken your slide out and this was all the way down, but you just forgot the 100x, I'm not gonna do that, right? I'm just gonna be like, hey, be sure you don't do that. Whatever. But this should be all the way down and on the smallest one. And if you forget once or twice, it's not a big deal. But if you're doing it consistently, you might get in trouble.
How would you bring it up to the highest power?
We'll walk through that when we go on. That's a good question. That one is Wednesday, next Wednesday. So we'll walk through all of that. Don't worry, because I know that most of you guys probably either haven't been using it correctly, or you have not used a microscope, or you're just not sure on the procedure. It's been a while, whatever. On Wednesday, we'll walk through exactly how to work up to that highest power. Correct. Okay. We'll talk about that. Good question. So that's the microscope. I want to bring that up today, and I will almost certainly talk about it one more time on microscope day, just because that is such a big deal. Okay. So if I was done with this and I wasn't saving this, this would go in the sharps container. Sharps container is the big plastic box looking red thing up here. About pick right. That's all I mean. Anyways, it's this thing here. So glass goes in here, your slides and any broken like tubes, they'll go in here. Your swabs should not be going in here. Your paper towels should not be going in here. So this should be glass. There's already crap that doesn't belong in here. That's why I'm bringing it up. Glass goes in here. Okay. You're done with your slide. We're not keeping it, and I'll let you know if I want to keep your slide. Okay. But if I'm not keeping it, then you will toss it in here. Please don't bring your things home with you. Anyways, this is your bio trash, and yeah, I'm usually gonna have it just a bag sitting out here. It doesn't usually stay out here like this. I usually tie this up at the end of the day. So obviously there's a lab right before you guys, and then you guys run right into the next lab. So when you're done, bio trash goes in here, and then I'll tie it up and it'll go in that container over there. So if I've already tied it up for some reason, like I'm hustling or something, and we're quite done, then let me know, and I will get the bag open for you again. Don't just like throw it somewhere or in regular trash. If it's touched actual bacteria, bio trash. It's not glass. Okay. If it's touched bacteria, bio trash. All right. All right, that's my husband. Anyways, yeah, okay. Any questions about the lab procedures? Like I said, we'll go through each of those procedures as we do them. Let's go over what we're going to be doing today, and then when we're done with that, you guys have to do that, okay? So for today, you have at each table, it's already open, that's okay. But to handle these appropriately and keep your swabs sterile, it should only be open part of the way. Then you will grab a swab by the stick end and don't touch that cotton end, right? Just like if you were doing swabs for COVID or something like that. All right, keep that clean. Then you can pick any surface you want in the lab. You may wet your swab with a little bit of water if you'd like, but that would help. Or not, I don't really care. But swab any surface in the lab that's non-living. Please do not swab each other's faces. Don't swab hands or body parts. I recommend bottoms of shoes. Easily. The number one grossest surface in this entire lab will be the bottom of your shoes. and that wasn't even included. Guaranteed it has more bacteria on it than anything else in the lab. The bottom of your shoes. That's literally why Japanese people take their shoes off in the house. Not just a cultural thing. It's literally your shoes are gross. So one of the most clean surfaces in a building will be a toilet seat because that gets cleaned often. They're not, and your butt itself doesn't actually have usually that much bacteria on it. Not the butt itself, right? Inside the toilet might be a different story, but anyways. So you swab whatever surface you want to, just non-living, okay? Shoes, even inside of your shoes, okay? There's nothing that's like on a person, please. Keep it safe. I don't know if you guys know this. The whole reason I'm even wearing, the only reason I'm wearing a mask today is because flu's going around. I don't want y'all getting any flu. Like it's hot right now. Like people are getting it left and right. If you don't know this, people getting flu real bad right now. And so I'm trying to protect myself with an N95 mask. I recommend that you do the same. Anyway, so I'm going to swab this gas thing here because I don't ever disinfect these. Think about other things too, like my keyboard, for example. You're welcome to swab that if you want. Or like these drawers. Like you guys know, we're going to disinfect this countertop before and after the lab. Do we disinfect these handles on these drawers? I don't even wipe them down either. So that might be a good one. You know, the stools or the floor itself, whatever you like to do, okay? But you will rub it down pretty good, whatever, maybe a inch by inch square area with your little swab, get a good swabbing of it. You may get it wet first if you'd like to. And then just zhuzh it on gently on this auger. Now you'll notice the auger is about the consistency of Jello. So don't jam it on there, just gently across the surface of the auger, just zigzag it on there. And you'll be done with that. Now I recommend also you need to label your plate. Always label your plate on the auger side, not the lid. Always the auger side, because that lid might get mixed up with somebody else. Also, there will be times when we're going to be doing different things in different parts of the plate. And if your lid turns, all of a sudden you don't know which part is which. Okay. Always write on the actual auger part of the plate. So I'm going to write my name, Lauren Y. I'm going to write my section. You can write 20749. Or you can just write 49, or you can write hybrid. I don't care. So you're probably going to remember that's hybrid. Write hybrid. You may write your table number, for example. So I'm not at a table, so I'm going to just put table 0. These things will just help you kind of organize what goes where. And then whenever I get the plates out from the fridge from a weather day or something, which is probably coming, right? That'll keep me organized with that. So write what you need to, but the bare minimum is your name and your section. That way, section is going to help me keep it organized if I need to move it around. Name is just going to tell you that it's your plate. So keep that on there. Anyways, I write around the edge because that way you can see what's going on inside of here when we start having colonies and things like that. So I write around the edge, right on the auger side. You'll notice too on your plates that the lid might have condensation all over it. That's just water from this being hot when we poured them and we cooled them. Always handle your plates with the lid down. Just because that condensation can drip down. And when we're trying to isolate our colonies and get colony separation, you don't want that water causing all to run together. So always lid down unless I tell you otherwise. Bottoms up, just like your drink, okay? Bottoms up, right on the actual auger side. And when you're ready to put this in the incubator, you can stack your whole tables up and put your whole stack in there, or you may put them individually, but it's your responsibility to put it in the incubator. So lid down, so it's auger side up. Auger out. And look at you guys. Right there. Okay. That are in here. I will check that. You guys have that advantage too. But yeah, you want auger up, and you put it on your thing over here. So I put mine in here, so you guys can stack yours up, move the whole table all together if you like. So you can get it all up together next time. And make sure you close this. All right. That is it. When you guys are done with your swab, if you swab a pretty normal surface that we're touching all the time, it's fine to put it into the regular trash. If you think that your swab is particularly nasty, then you are welcome to put it in biotrash. Okay. Close up. You biotrash gets this orange back. And then when you guys are done with that, you're free to go. Don't forget to clean your tabletop and wash your hands. I just don't want you to slip on that, you know? I don't know where that came from.
Go back to the car. I appreciate that. I was so scared. I didn't know how long it would take me to walk.
That would be a pain.
Yeah, I just started walking day before yesterday.
As long as they let me go in. Then I'm so bad. Not like I could want to.
Okay, sounds good.
We don't know who the professor is.
No, could you show me?
Very off, Adam's family.
Oh, okay.
The uncle with the bald hair.
Yeah. No, Marie Davis. Good name. Yeah, right. It almost never gets misspelled. Okay.